but many contained a dark green, almost black mold. Under a microscope, Sorensen noticed that the samples from the Cleveland homes were structurally similar to the mold which killed the livestock in Europe. It was, in fact, Stachybotrys atra. Investigators now knew that spores from Stachybotrys atra were in the air of the sick babies' homes. But they needed to know whether the Stachybotrys spores in Cleveland contained the same toxins as those which killed the livestock in Europe. To find out, scientists grew the spores found in the Cleveland homes in a mixture of rice. Stachybotrys grows well in rice because of the moisture in the grains. The mold was then ground and mixed with a series of solvents to extract the chemicals contained in the spores. Using what is called high-performance liquid chromatography, the physical characteristics of the spore chemicals were analyzed, and scientists discovered that the Cleveland spores were identical to the highly toxic mycotoxins that killed the livestock in Eastern Europe. They were a special class of mycotoxins called trichothecenes.
The trichothecene mycotoxins are among the most potent protein synthesis inhibitors known. By that, I mean they disrupt the ability of cells to make proteins, and without that ability, a cell will die.
When scientists searched the homes of the sick children, they found the source of the Stachybotrys spores in mold found growing in the basements. The mold was found in all but one of the sick babies' homes. But Stachybotrys was also found in half the homes tested where healthy children lived. If it was Stachybotrys that caused the death of one Cleveland infant and the serious illness of 9 others, why didn't it affect all of the infants who lived in homes where it was found? Spores from the Stachybotrys atra mold were found in all but one of the sick infants' homes, but were also found in half of the homes of healthy children living in the same neighborhood. Scientists wanted to know why some children developed lung bleeding while others did not. They soon discovered that the number of airborne spores in the sick babies' homes were far higher than in the homes of healthy children. In effect, the homes of sick babies contained more spores and more poison.
Stachybotrys is generally not considered one of the major components in indoor air, major fungal components.